Good afternoon now. It is Tuesday, November 9th. We are on to our second point, halfway through our urinary system material. So we'll finish day 2 today, and then day 3 on Tuesday the 16th. That is because Thursday is a holiday, so Veterans Day. So enjoy that. Take that time to study and be prepared for next week when we have a Tuesday lecture. Always a fun thing with a Thursday exam. New information 2 days before the exam. So again, you should use Thursday to look at that material. You should, you've got the lecture slides, you have previous lectures, you could look at all those things to help you to be successful. So hopefully it's not too much new information on Tuesday. You do have 2 assignments due on Tuesday, Unit 26 review, and then also PhysioX Exercise 9, which has 6 activities, which, as you know, is not hard, but can be time consuming. All leading up till next Thursday's lab and lecture exam. Again, you've already taken 3 of these, so you know what to expect from that. The other thing you know to expect from a Thursday exam is assignments due on Tuesday when you come back. I can't have you go whole 5 days without thinking about anatomy and physiology. So there are 2 things that I'm going to have you do to help you to be successful when we come back. And that is the pre-lab on the reproductive system, our last organ system we have to cover. And we will be doing a lot of talking on Tuesday the 23rd about meiosis. And meiosis relates very, very strongly to mitosis. And from previous experience, I find that students do not have difficulty recalling mitosis from way back at the beginning of 430. So by having this labster due when you come back, even if you don't understand all the meiosis stuff, if it reminds you and recalls the mitosis stuff, that will help you to be successful when we talk about it on Tuesday. Then you get another holiday to eat turkey and impress all of your family with all the knowledge you have about the digestive system from last month. Impress your family by talking about deglutition and all of those fun things, and they'll all be impressed. And since they're in person, they will give you $20 and not have to ship it in the mail. All right, then we come back, 3 more lectures, and then it's all exams and we're done. Holidays. All right, so that's the game plan. We're rapidly approaching the end. Questions on any of that? Excellent. You know how much I enjoy my stunned silences first thing at the beginning of class. Spectacular. All right, so let's actually stay here. We left off last time and we talked about the anatomy of the nephron. The nephron is basically the structural and functional unit of the kidney. And it is made up of 2 types of structures. What are the 2 types of structures that make up a nephron?
Tubules.
Excellent. And what's the other one?
Collecting duct.
Well, true, but remember the collecting duct is actually not a part of the nephron. It is definitely there in the kidney, but it isn't a part of the nephron. Nephron is made up of tubules and what other type of structures?
Blood vessels.
Blood vessels, there you go. And I like that you guys are remembering some of the things that this is made up of. I'm going to simplify things a little bit here. Rather than have a normal shaped nephron, I'm going to straighten it out. And with the magic of whiteboard, I can make that a little taller. All righty. Nope. And now that I've done that, I've changed my mind. Let's move this down. And that down. So here are all of the tubular structures that make up a nephron. What are the components? What are the 4 tubular structures that make up a nephron?
Proximal convoluted tubule.
Okay, excellent. We have the proximal convoluted tubule. Which is proximal to what?
The renal corpuscle.
Renal corpuscle, excellent. And what is the tubular component of the renal corpuscle?
The glomerulus.
Close. The glomerulus is the blood vessel part of it. So we could call it the glomerular capsule. Or what was the other name we could give to it? Bowman's. That's okay. No R, just Bowman's. Although you do get a letter, so that's good. Excellent. So glomerular capsule or Bowman's capsule. Excellent. We're halfway there. What else?
The loop of Henle.
Loop of Henle. After good old Bob Henle, or again, we could also call it the Nephron loop. Excellent. Which leads into what?
Distal tubule.
There you go. Don't forget, it's all twisted around, so it's the distal convoluted. Tubule. Excellent. And that is the nephron. And we will cheat a little bit and let's change colors of it. Because we also know there is going to be a little processing that'll take place, as someone mentioned earlier, in the collecting ducts. The collecting duct, remember, is not a part of the nephron, but it does process the filtrate. So we'll add it to the end of our tubule here, even if we're changing the color of it. And of course, as we mentioned, when the filtrate that is first produced in this then leaves, let's go back to black. When that fluid and stuff leaves the collecting duct into the minor calyx, what does it form?
Filtrate.
It's filtrate when it comes out of the glomerulus in the glomerular capsule when it's getting processed. But once it leaves the collecting duct, is it processed anymore?
No.
No. So there you go. Ruby's got it. It is now going to be our urine into the urinary tract, where it then is stored until a socially appropriate location and it can be left. However, as you guys have also mentioned, there are blood vessels involved in this process as well. So let's draw some of those. Again, we'll cheat with purple. We have the first blood vessel that feeds into that renal corpuscle that technically isn't part of the nephron. And what was that blood vessel that feeds into the glomerulus that isn't part of the nephron?
Afferent arteriole.
Excellent. Our afferent arteriole that then indeed feeds into our glomerulus, which feeds out into what blood vessel?
Efferent arteriole.
Excellent. You got you beat just by a little bit, Ariel, but excellent. Thank you. Excellent. Into the efferent arteriole. Well, he had the advantage of speaking, you had to type. So, and if you type like me, although nobody types like me, I'm a horrible typer. That efferent arteriole feeds into what? Excellent. Peritubular capillaries. And what other structure besides the peritubular capillaries could be here on, say, 15% of the nephrons? There we go, Amy. Vasa recta. Perfect. Although I am going to cheat a little bit. Move this down. Move this down. Oops. Which means I have to move this down. I like that. I can't do that on the whiteboard. I can't move things around when we're in the classroom. One of the few advantages of being here online. Excellent. Then when it leaves, oh, let's do this. The blood then enters into a venule where it leaves the nephron. And what would we say about the state of the blood as it leaves the nephron?
Oxygen poor.
True, absolutely. It is going to be oxygen poor. Because what was all that oxygen used to do?
Supply the tubules and everything.
Yeah, exactly, to do the work. And what is the work of the nephron?
To filter the blood.
Yeah, cleaning the blood. All right, so we have that clean blood, oxygen poor, clean blood that leaves. Whereas up here in our afferent arteriole, we have that dirty blood that entered. All right, so our dirty blood, which, as we know, came from the abdominal aorta, came from the left ventricle. So let's be consistent, as we were talking about before. Since it is systemic arterial blood, it is going to be oxygen rich. And that oxygen is going to be used during the process to process that blood, clean the blood, produce the urine, and then that oxygen poor, clean blood goes back to the inferior vena cava, goes back to the right side of the heart to go to the lungs and get oxygen, and then it's on its merry way. And that's our job. We've talked here, we have a simple version of our anatomy. And as we just finished talking about, our goal is to process the blood. And this processing of the blood is a three-step process. The first of those steps is what is known as glomerular filtration. All right, this is step one. And actually, let's put a 1 in front of it. Bingo. Step 1, glomerular filtration. Now, there is a lot in a name. Where do you think glomerular filtration takes place?
Near the glomerulus.
Yeah, in the renal corpuscle. Out of the glomerulus, right? It occurs in the renal corpuscle. And with a name like filtration, what direction do you think it's coming? Into the blood or out of the blood?
Out of the blood.
Yeah. Remember, we talked about how that glomerulus is very, very leaky. And so we have that water and stuff dissolved in the water, part of the plasma coming out. It comes out of the blood. And as the name indicates, this is how we form our filtrate. And again, remember, as we talked about, we produce over 200 liters of that during the course of a day. Of course, we don't produce 200 liters of urine, so it still needs to be processed. Now, let's think one more thing. As we've talked about, this is a fenestrated capillary. It has those podocytes on it. Do you think this is a passive or an active process?
Passive.
Yeah, absolutely. And of course, what does that mean?
Does not use doesn't use ATP to
Excellent.
transport.
Which is really good. We're not using ATP. However, the problem with not using ATP is we can't be selective. We can't decide what we are going to pull out of the blood in this case. In this case, basically all we're doing is sorting by size. The example I always like to think of this is after I come home from a long weekend, right? You've got the holiday off, it's 5 days off. So it's a perfect opportunity to fly to France for a few days. And when you get back from France, you have that huge stack of mail. And what's the first thing you do with that huge stack of mail? Come on, someone must have flown to France. Yeah, sort it. And typically you sort it by size. All the small envelopes on one side, all the big stuff on the other. right? Why do you do that? Nobody's been away from, I know COVID has changed things, but nobody's been away from their house for more than a day now where they don't have to sort their mail. Why do you sort your mail by size?
Sort by order of importance.
Right. Typically the small envelopes, those are the things like the bills or the checks or the important letters, things along those lines. Whereas the big stuff are the coupons and the mailers and the junk mail. However, does that mean you take the big stack and just toss it without even looking at it? No, of course not. There could be some important things, right? Your acceptance to your nursing program could be in there. Those portraits that you ordered could be in there. And by the same token, is everything in a small envelope going to be something important? Or can some of those small envelopes be about the importance of getting homeowners insurance when you're clearly living in an apartment, right? So we're sorting by size, but notice when we sort by size, anything small comes out. And that's going to be both good and bad stuff. And again, we'll put that in quotes. Right. The reason we put good and bad in quotes is, as we've learned, it's not all that straightforward. Like carbon dioxide, as we learned in the last section. Is carbon dioxide a good thing or a bad thing?
Bad thing.
Well, it is a bad thing. Does that mean we want to get rid of all of it that we possibly can out of our blood?
No.
No, because it plays an important role in pH. So notice we definitely don't want too much CO2, but at the same time, we don't want to get rid of all of it either, right? So while it's a bad thing, typically, we don't necessarily want to get rid of all of it. Some of it is actually good for regulating pH. So again, what we think of as good and bad can be somewhat subjective. But the point is, we can't be selective. All right. Questions on glomerular filtration? All right, and as we mentioned, during the course of a 24-hour period of time, whoops, no, hold on, I want that to stay black. But here, as I mentioned, we produce 200 liters of that filtrate here in our renal corpuscle. But we don't want to produce 200 liters of urine. How much urine do we produce during the course of the day? I did ask that question out loud, right?
Yeah, like 1 to 2.
Yeah, there you go. I'll see, I should have used yellow. That's 1 to 2 liters of urine during the course of the day. So clearly glomerular filtration can't be all. There are 2 additional steps. The next step in this process is what is known as tubular reabsorption. Again, reabsorption tells us a lot. If you think about this, because this loop of Henle, this distal convoluted tubule, the collecting ducts, connect to the minor calyx, connect to the ureter, connect to the bladder, connect to the urethra, are outside the body. So anything in this tube is considered outside the body. And if it stays in this tube, it's going to leave the body. So anything we want to keep, we got to get it out of that tubule, back into the interstitial fluid. And from that interstitial fluid, we've got to get it back into the blood. So notice with tubular reabsorption, we are moving from the tubule into the peritubular and vasa recta capillaries. So this one is moving into the blood, and more specifically, let's say out of the filtrate and into the blood. And Where do you think tubular reabsorption occurs? Absolutely, that's a great way to describe it. Technically, it occurs throughout the tubules. So that means the proximal convoluted tubule, the distal convoluted tubule, the loop of Henle. Oops. And even the collecting ducts that we talked about. However, while it occurs throughout the tubules, you are absolutely correct, Ariel, that most occurs in the proximal convoluted tubule. So most of it occurs in the proximal convoluted tubule, but it occurs throughout this process. So yes, the majority of it is happening here. 5 there. We'll put 4, 3 here, 4 there. We'll put 3 or 4 here. We'll even put a couple in the collecting duct, but we have all of that tubular reabsorption. Coming back, and I put too many in the other area, so let's put a couple more here. Perfect. Now, do we want to let just any old thing back into the blood? No. So this is definitely going to be an active process. Yes, it's going to use ATP, or mostly. We know there's always exceptions. Mostly uses ATP, but the advantage of this is we can be selective. And so in this case, what are the things we want to bring back?
Water.
Absolutely. What else?
Sodium, potassium, chloride.
Ions, absolutely. What else?
Maybe glucose.
Absolutely, things like glucose,
Calcium.
calcium, although I think calcium falls under enzymes, but we could put, well, let's leave it under enzymes. Stuff like that. Basically our quote unquote good stuff. Right? All the stuff that the body or needs, all the things that we want to maintain in the blood. This is where we bring the good stuff back. All right. Questions on that? Excellent. All right. There is a third process, and this third process is tubular secretion. Now, where do you think tubular secretion occurs? And this time I'll change the color. Let's use green. I haven't used green yet.
Throughout the tubules.
Exactly. So again, that means the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, even the collecting duct. And do you think it happens equally through all of them? No. Most occurs where? Collecting duct is indeed a good guess. But if you remember, we said the collecting duct and the distal convoluted tubules are where we're fine-tuning. Tubular secretion isn't quite a fine-tuning type of event. So in fact, this also mostly occurs in the proximal convoluted tubule. Notice I purposely didn't number tubular reabsorption 2. Yes, glomerular filtration has to happen first. We have to form the filtrate before we can process the filtrate. But once we have the filtrate, we don't take all of the things out of it that we want and then put all of the things that we don't need back into it. So tubular reabsorption and tubular secretion actually are both occurring at the same time. So if anything, we could call them 2A and 2B, but really it's not a sequential event. Obviously, glomerular filtration has to happen first, but tubular reabsorption and tubular secretion occur at the same time. Now, what is the direction of tubular secretion? We want to secrete something outside of our body. Which direction is that going to go?
Into the,
Into what?
I'm not sure, into the blood.
Well,
What
so
if-
again, remember, we want to secrete it. We want to get rid of it. We want to send it to the outside of the body. Where's the outside of the body?
Interstitial fluid.
True. And from the interstitial fluid, then into the tubule. So you absolutely have the right idea. We want to go out of the blood and into the, oops, wow, this is way too big. Out of the blood and into the tubule. So in this case, and again, we change the color just to make sure that we emphasize the point. Most of it again occurs here in the proximal convoluted tubule, where things are moving out of the blood and into the tubule. Now, let's cheat and do this first. What kind of stuff do you think we want to get rid of?
Nitrogen.
Yeah. Nitrogen-based wastes. What else?
Ammonium.
Yep, that's a nitrogen-based waste, absolutely. Biological metabolic wastes. What else? What else don't you want in your body?
Urea.
Sure, which again is another nitrogen-based waste. These are all metabolic waste. Excellent. But what about those pesticides on your apple that you didn't wash off? What about excess hormones that you have floating around in your body? What about excess ions? What about those hydrogen ions if we want to maintain pH? Basically, if you think about it, we want to get rid of the quote unquote bad stuff. And if we are going to be selective in getting rid of the bad stuff, is this mostly going to be an active or a passive process? There we go, active. Oops. All right, so again, it's going to mostly use ATP. There is going to be a little bit of passive, as we'll see. And again, the advantage of using ATP is that it allows us to be selective. We don't have to just randomly kick things out of the blood. We can specifically kick things out of the blood that we want out of the blood. And so after all three of these processes are complete, that is when we then have our urine and our clean blood. All right. Questions on that? I've done it here with my amazing drawing, but notice I totally cheated and took this from your textbook. Here, like I did, they have this nice simplified version of a nephron to emphasize again these three steps. we have that glomerular filtration that is taking place in the renal corpuscle, right? The glomerulus, the parietal layer of Bowman's capsule and Bowman's space. Right? This is where we basically sort by size. That passive formation of filtrate, pushing things out of the glomerulus to make those 200 liters of filtrate. Then within the tubules, we need to bring over 99% of that stuff back into the blood. That is the process of tubular reabsorption. Again, that mostly is going to occur in the proximal convoluted tubule, but it will occur throughout the entire tubular structure. It's active, it's selective, and we bring back the stuff that we want. Lastly, and again, occurring at the same time, remember, even though these are numbered 2 and 3, we don't do all of the reabsorption and then all of the secretion. These are occurring at the same time. And so with that tubular secretion, again, most of it is occurring in the proximal convoluted tubule. But this is when we're able to move big stuff, either big stuff that we weren't able to sneak out here in the glomerulus, or maybe some small bad stuff that snuck its way back in. We then have to kick it out, get it into the tubule so that it becomes part of our urine. And so these are the 3 processes we need to make sense of over the next day and a half. All right, questions on that? All right, then let us start with step 1, glomerular filtration. Again, this is where we form the filtrate. This is where we're moving out of the blood. And again, this is a passive process. And again, in the process, we're producing filtrate, and a lot of it. Filtrate is basically blood plasma with all the little stuff in it, right? And both the good stuff and the little stuff, just small stuff in it. No formed elements. So no red blood cells, no white blood cells, not even those thrombocytes should make it out into the Bowman's capsule. And most of the large proteins, fibrinogen and things like that, are going to be kept in the blood plasma. So it's basically just blood plasma and all the good and the bad small stuff. However, we're going to reabsorb 99+% of that. So what is actually released from our body, voided from our body as urine, is pretty much a small amount of water, metabolic waste, and any unneeded substances. Again, I can't help but emphasize this point a lot. Again, we've talked about it a little bit in the last class, but there are a lot of homeostatic processes involving urine, all right? From things as silly as if you get stung with a jellyfish, you urinate on the sting and it doesn't hurt as much. There is some value to that, but things like vinegar and other things could be better. And that's not horrible. I've heard of people urinating on their feet to help with athlete's foot, which I promise you does nothing. But the really bad ones are the people who wash their face with their first urine of the morning, or they drink their first urine of the morning because it's supposed to have some kind of mystical, magical, homeostatic process to it. But again, remember what urine is. Urine is basically all of the things that your body didn't want. So there's no point in putting them back into yourself again. All right, so don't wash your face and drink urine. All right, let's talk about the process of making that filtrate. We talked about the anatomy of this last time, and notice here we have that pretty picture that we talked about last time. Notice we have our glomerulus, that fenestrated capillary with all the little holes in it, just a single endothelium, a simple squamous epithelial tissues, poked holes in like Swiss cheese. Its basement membrane, and then those specialized foot-like processes of our podocyte. As our blood plasma travels through the glomerulus, about 10% of it is filtered out. And as I keep mentioning, you produce about 200 liters of filtrate during the course of the day, which gets processed down to 1 to 2 liters of urine. As we talked about, this is an incredibly thin membrane. Remember, this is a passive process, so does not use ATP. So there are things that we want to do to enhance it, to make it more efficient. One of those is that thin membrane of our urinary membrane. With 1 million nephrons, we have a huge surface area. And again, that's 1 million per kidney. We have a huge surface area for this. But remember, one of the things that we said about the glomerulus is that it has a very high glomerular blood pressure. And that is going to help to force that filtrate out of the capillary. We can actually measure the amount that is made. And again, real quickly, we did the anatomy of this last time, but I want to look at the pretty picture from your textbook. Remember, our filtration membrane is made up of that fenestrated endothelium of the capillary. It is made up of those podocytes and their foot-like processes with their little filtration slits in between. And like I said, that fused basement membrane, what is known as a dense layer. Here's the illustration that we saw before, one very similar to it, but this is the picture from your textbook, which I really, really like. Here, using that electron microscopy, using that electron microscope, we can actually see the anatomy of this. We can clearly see, here is our endothelium, and we can see those fenestrations in that fenestrated capillary. And here we see the foot-like processes of the podocytes and the little slits in between. So if you are water, or if you're some solute that's dissolved in water, basically you need to be able to wiggle your way through these spots to come out. So it's like using basically a sifter. We are just selecting from size. You put your flour, you shift it, and all the individual pieces of flour come out, but all the big chunks stay behind. And that's what we're doing. We're just sorting by size. Now, luckily, we learned way back in the cardiovascular system that we could do some math to determine how things move in relation to a capillary. Remember, as we talked about, there were 4 pressures that determine how things move in a capillary. I don't know if they're all going to fit if I leave it this size, so let's make it a little bit smaller. There's the hydrostatic pressure of the capillary. And remind me, was that an outward force or an inward force?
Outward.
Outward force, excellent. So we'll just put an O there. There is the hydrostatic pressure of the, in this case, it was the interstitial fluid. And was that an outward or an inward force?
Inward.
Inward, excellent. There was the osmotic pressure of the capillary or the blood. Was that an inward or an outward force?
Inward.
Inward force. And then, of course, there was the osmotic pressure of the interstitial fluid. And was that outward or inward?
Outward.
Outward. Excellent. So we can deal with these exact same pressures here in the glomerulus as well, with one big change. It's not going to be the interstitial fluid that is going to be the force here. Oh, I messed myself up by doing this, but that's okay. It is now going to be the hydrostatic pressure of Bowman's capsule or of the filtrate? So as water comes pushing out of the glomerulus, we have that Bowman's capsule right next to it, where that water is going to bounce off of it and want to move back into the capillary. Someone remind me again, what is an osmotic pressure? What do we mean when we say osmotic pressure? Hydrostatic pressure is the force of water pushing on its boundaries. What was osmotic pressure?
Was it the force inside of the red blood cells?
Kind of, it was close to that. Not just the red blood cells, but all the stuff inside of the blood or inside of the interstitial fluid. Remember, that stuff attracts water. So remember, osmotic pressure is the pressure of stuff drawing water to it. All right, so far we good with that? We remember that? We good with that? So let's think about this. In Bowman's capsule, after the filtrate comes off, does the filtrate stay in Bowman's capsule?
No.
No, it leaves. How does it leave? How does the filtrate leave Bowman's capsule?
Through filtration.
Well, it came out of the glomerulus into Bowman's capsule, but
Right.
Bowman's capsule is a cup that collects the filtrate, and it leaves that cup to go into what?
Proximal convoluted tubule.
Proximal convoluted tubule. Excellent. So if the filtrate, as soon as it's made, leaves into the proximal convoluted tubule, does any of that filtrate stay behind to attract water to it? Well, I'm asking the question, so what's the obvious answer?
No.
No, exactly. So if we want to measure the pressures that are affecting the glomerulus, if we want to measure the pressures that are determining the rate of filtration, if we want to calculate the net filtration pressure, there are only 3 pressures we need for this. Let's go to the whiteboard to do this. Excellent. Our goal is to calculate the net filtration pressure. So let's do our drawing here. Here we have, and I'll just draw it as a box, our glomerulus that is going to be surrounded by Bowman's capsule. As we just mentioned, here inside of the glomerulus, we have a hydrostatic pressure of the capillary. And remind me again, did we say that was an outward force or an inward force?
Outward.
Outward force, excellent. In here, we also have the osmotic pressure of the capillary. And did we say that was an outward or an inward force? Excellent, thank you, Amy, inward. Lastly, out here, because of close proximity, we also have the hydrostatic. Let's say it that way. The hydrostatic pressure of Bowman's capsule. And is this one an outward or an inward force? Excellent. Also an inward force. So notice we have 2 inward forces: the hydrostatic pressure of Bowman's capsule, the osmotic pressure of the caps of the glomerulus. Maybe we should say glomerulus because capillary and capsule is too confusing. So let's change this to glomerulus. And this to glomerulus. Excellent. Oops, that should have been blue, but that's all right. Oh. All right, I lied. It does bother me. Excellent. One outward pressure, two inward pressures. So of course, how would we calculate our net filtration pressure? How would we figure out what the net pressure would be? Any idea? I'll give you a hint. We take the outward force, and what would we do to that?
Add them together.
Well, but remember, notice these are opposing forces. So when we have opposing forces, do we necessarily want to add them together?
We subtract them.
Subtract them. So it's our outward forces minus the inward forces. In other words, our net filtration pressure would equal the hydrostatic pressure of the glomerulus minus the osmotic pressure of the glomerulus plus the hydrostatic pressure of the Bowman's capsule. So now all we need are numbers. We talked about this in the last class. In a typical capillary, what is the range of pressures? What was it at the arterial end and the venous end? Does anybody remember? I know we talked about it just in the last class. Does anybody remember in a typical capillary?
35 millimeters of mercury.
Excellent. And at the venous end?
17.
Remember, we said this glomerulus is a very high pressure capillary. And sure enough, the hydrostatic pressure of the glomerulus, that pressure inside of it is 55 millimeters of mercury. All right. Questions on that? Nope. What just happened? Sorry. Hold on. Oops. There we go. Sorry, I lost my whiteboard. All right. So we have that. Now we need our other pressures. Can we all see this now?
Yes.
All right. The capillary, remember, has a lot of stuff inside of it with that blood. So not surprisingly, the osmotic pressure of our glomerulus is relatively high as well. And that is about 30 millimeters of mercury. If you remember, in a typical capillary, it was 25. So this is a little bit higher because this is the quote unquote dirty blood, but it's similar. 35 to 55, that's a huge difference. So it's a very, very high pressure capillary. And the osmotic pressure is a little bit high as well. So what we're left with is this hydrostatic pressure of Bowman's capsule. Now, again, remember, this is the force of water pushing it on its boundaries. If you're washing dishes, and as the water is coming out of the faucet, you took a plate, and you took that plate and put it right up against the faucet as all that water was coming out, what would happen?
Shoot out everywhere.
Yeah, it would spray out all over the place. So this is so tight that as the filtrate is coming out, it's really pushing on the boundaries. So this hydrostatic pressure of Bowman's capsule is pretty high as well. And the hydrostatic pressure of Bowman's capsule is equal to about 15 millimeters of mercury. So now that we've done all that, can we come back here and actually calculate what our net filtration pressure is going to be?
Yep.
55 minus 30 plus 15, which equals what?
Positive 10.
And of course, as a positive number, is that an inward force or an outward force?
Outward.
And so that positive 10 millimeters of mercury outward force is what keeps our filtering process going. Now think about that for a minute. When we are in your, if you were measuring your systemic blood pressure, all right, as I mentioned, I just had my appointment this morning. I get checked out every week. And as usual, my blood pressure is at a very solid 315 over 225, right where I like it. And if it fluctuates 5 millimeters of mercury as I sit up or as I lay down, is that a big deal? A 5 millimeter mercury change systemically throughout my body?
At those numbers, probably not.
Okay, true. Even at 120 over 80 is a 5 millimeter change in mercury. I mean, millimeters of mercury change, a significant changes. If you were at the doctor and yesterday it was 120 over 80 and tomorrow it's 125 over 80, are they rushing you to the emergency room?
No.
No, that is a very minor, almost insignificant change. However, notice here in the glomerulus, if as small as a 5 millimeter of mercury change occurred here, if suddenly your net filtration pressure went from 10 millimeters of mercury to 5, is that going to be a big deal?
Yes.
Yes. So notice this is a relatively small number. And so one of the things we have to work really, really hard at is to maintain this number so that we can maintain constant filtering of the blood, constant cleaning of the blood, and maintain the overall health of the body. So this relatively small difference can have a huge impact on how the kidneys function. All right, we've done it with my pretty pictures. Let's look at the pretty pictures from your textbook. Again, we have our glomerular hydrostatic pressure, 55 millimeters of mercury outward. We have our capsular hydrostatic pressure, 15 millimeters of mercury. And as we said, that's an inward force. So again, this is out, this is in. And our osmotic pressure of the blood is 30. And again, this is an inward force. So to calculate those, we take our outward pressure, we subtract the inward pressures. And as we mentioned, and as the pretty picture in your book shows us, our net filtration pressure is at an average of 10 millimeters of mercury. And it is an outward force, meaning that blood plasma and all the small stuff come out. And they come out at what we call the glomerular filtration rate. Glomerular filtration rate is about 125 milliliters per minute in males, about 105 milliliters per minute in females. And of course, why is there that difference? Why is there a difference between males and females?
Reproductive system?
Not a bad guess. I would say it's okay. First, is it statistically different? Is it significant? Statistically, mathematically, it may be different, but is it clinically significantly different?
No.
No, of course not. My guess is it's because males tend to be slightly better. I mean, bigger than females, right? So yeah, it's probably related to size. Males' livers, exactly. Hannah's got it. It's the difference in size. Males are slightly larger, so they have slightly bigger kidneys, so they make slightly more filtrate. But again, it's not as significant from a clinical standpoint. However, what we did talk about is how important it is to make the filtrate at an appropriate rate. Remind me what happens if we make too much filtrate. If we're making too much filtrate, if the filtrate is too high, what was the problem with that?
It goes under processed.
Yeah, it is under processed. We won't have the time to process it properly. We won't have the time to filter it properly. Whereas if our filtration rate is too low, if we're not making enough, or not making the filtrate fast enough, or making too little of it, let's be consistent, making too little filtrate, what's the downside of that?
Over processed.
Yeah. It will be overprocessed. So maintaining that appropriate glomerular, net filtration pressure is going to help us to maintain an appropriate glomerular filtration rate. And clearly, this is something that has to be very, very closely monitored. And as we'll see, there are going to be 3 primary methods that we're going to use to do this. Any idea what those 3 methods might be? How do we regulate most of the organs in our body?
Hormonal regulation.
Hormonal, excellent. Oops. Hormonal regulation.
Sympathetic.
Yep, neural, but you are absolutely correct. As we already talked about, it is going to be the sympathetic nervous system. Regulation. So what's the third?
Ionic?
Not a bad guess, but think back to in the lungs. Did our nervous system communicate with every single individual alveoli?
No.
No. So how was each individual alveoli's perfusion and ventilation rate controlled?
Auto regulation.
Yeah, that had that intrinsic or auto regulation. And that's exactly what we're going to see here. We are going to see an intrinsic auto regulation. We're going to see a hormonal regulation, and we're going to see neural regulation. And as was pointed out, that is going to be surprisingly, although when we see it, it's going to totally make sense. It is by the sympathetic nervous system. All right. Questions on that? All right, that's going to be a lot to cover. So this is a good point to take our first break. Let's go ahead and take our first break. We'll take a 15-minute break. Let's see what time it is now. It is 1 o'clock. So let's restart at 1:15. And at 1:15, we will start the recording again. So any questions on this before we take our break? All righty, I will see you guys in 15 minutes. All right. Any questions before we get started? All right, let us talk then about regulation of our glomerular filtration rate. As we've talked about, we're dealing with a very small pressure that is playing an intricate role in producing the right amount of filtrate so that it does not get over processed, does not get under processed, and is properly cleaning the blood. And as we talked about, when you're dealing with 1 million nephrons per kidney, there has to be some type of intrinsic or auto-regulatory type of process. And in fact, as it turns out, there are two. The first one is what is known as the myogenic mechanism. Now, of course, myo refers to what?
Muscle.
Muscle, exactly. So again, our goal here, let's go to the whiteboard. Is to, let's make a box. Nope, not a box. So as we know, we have our glomerulus. And we know feeding into it is our afferent arteriole. And feeding out of it is our efferent arteriole. Yeah, look how pretty that is. Excellent. We know that we need to maintain the glomerular hydrostatic pressure inside of our glomerulus so that we can produce the appropriate amount of filtrate. However, as we just finished talking about earlier, systemically, our blood pressure is pretty dynamic. Whether I'm sitting up or whether I'm standing, whether I'm walking or whether I'm exercising, right? Just laying down and closing my eyes and opening my eyes. That something as subtle as that can cause dynamic changes to occur in our blood pressure. And so every time we sit up, every time we close our eyes, every time we think about eating a cheeseburger or think about the test coming up or some other kind of dynamic event occurs, do we want the mechanism of our kidneys to change how they're processing our blood? These are the easy questions, folks.
No.
No, of course not. Absolutely. And luckily, as we know, our afferent arteriole has those specialized cells, those juxtaglomerular cells or granular cells that are a combination of smooth muscle and endocrine cells. Oops. That are smooth muscle cells. And these smooth muscle cells respond to pressure changes. So what's special about these cells is that they respond to pressure changes. So let's cheat and move this up here. So let's think of a situation here. We want to maintain a consistent glomerular hydrostatic pressure. So let's take example 1, where we have an increase in our systemic blood pressure. So in this type of a situation now, we are getting an increased pressure, right? The force of the blood is pushing on our afferent arteriole. Now, of course, when we get this increase in pressure that pushes on the walls, what happens to our smooth muscle layer when that occurs? What happens to a blood vessel as the blood pressure increases? Well, what
Stretches.
happens? Yeah, it stretches. That blood pressure stretches. Inside of this are stretch receptors. And so we have these stretch receptors that when they stretch, they fire more action potentials. And those action potentials that are produced, they affect the smooth muscle. What would we want to do to the smooth muscle in this case to bring the blood pressure back down?
To dilate?
Exactly. It causes dilation, and that dilation decreases blood pressure. So that maintains the consistency. We had an increased blood pressure here, but our vessel dilates. And so as the vessel dilates, the blood coming into it drops the blood pressure and it maintains a consistent pressure. It lowers it so that it's the pressure we want, so we produce the right amount of filtrate. So let's do the opposite. Oops, why did that? All right, there we go. We have a decrease in systemic pressure. I'm now sitting in that yoga class, right? Sitting on the ground, or that meditation class, sitting on the ground, eyes closed, lotus position, oming. And so there's a decrease in systemic pressure. And so as there is less systemic pressure, as the blood pressure decreases, our vessels, remember, which have some elasticity, are going to recoil. So there's less stretch. And with less stress of the blood vessel, what happens to the firing rate of those stretch receptors?
They go down.
Exactly. Refire fewer action potentials. So our stretch receptors fire fewer action potentials. And as they fire fewer action potentials, what is the effect on the smooth muscle going to be?
It'll contract.
Going to cause a contraction. And as it contracts, as our blood vessel gets smaller, what happens to the blood pressure in it?
Increases.
And again, in that way, it compensates for the decrease in the systemic pressure, and it can help us to maintain that consistent pressure. Notice the goal is the same in both of these cases. No matter what's happening outside in the body, if you're sitting, if you're standing, if you're eating, if you're watching TV, if you're being scared, if you're sleepy, we want to maintain that consistent pressure. So regardless of which way this occurs, the blood vessel, the afferent arteriole, notice this is all occurring, and let's make sure we emphasize this. This all occurs in the afferent arteriole. So this myogenic mechanism is specifically occurring in that afferent arteriole in those juxtaglomerular cells, the granular cells, those smooth muscle cells. Excellent. Questions on this? Now, like I said, this can help us to compensate for whether we're standing or whether we're sitting, whether we're laying down, whether we're sleepy, whether we're scared. However, if there's something that makes our blood pressure spike, like a bear with the axe walking in the room, or us exercising, or something along those lines, are we going to be able to change the diameter of our afferent arteriole enough to compensate for something like that? What do you think? Microphone is on, right?
Yeah,
Okay. So do you think that major changes in blood pressure will be able to be compensated for in this way?
no,
Maybe not.
No.
Yeah, bear with an axe takes a bite out of your arm and you lose 2 pints of blood. But that's okay. You can keep your kidneys going because your afferent arteriole is going to squeeze it. Is that going to happen?
No.
No, of course not. So again, normal changes, normal everyday changes of that dynamic change in blood pressure, like I said, as you sit, as you stand, as you get called on in class, right? All of those things, those small changes, this can compensate for. But we need more than this. So we have this myogenic mechanism, again, an intrinsic one. And the beauty of this is, again, all 2 million nephrons all have an afferent arteriole. So we can constantly be changing the pressures. However, as we talked about in the last class, for every single nephron is having that glomerular filtration, that glomerular hydrostatic pressure at 55 millimeters of mercury. Is that really going to always produce the perfect amount of filtrate in every single nephron? I mean, is every single nephron in our body the exact same length and the exact same size and works exactly the same way?
No.
no,
No. So remember, as we talked about in the last class, we have that juxtaglomerular apparatus, that quality control, where each one of our nephrons has that quality control process, that juxtaglomerular apparatus. And that is our second intrinsic mechanism, what we call the tubuloglomerular feedback, right? If you think about it, it starts in the tubes in the distal convoluted tubule, and it tells the glomerulus, it gives the glomerulus feedback on how well we're doing at processing our filtrate. Remind me again. So our tubular glomerular feedback uses the juxtaglomerular apparatus. Right, just say that to grandma on Thanksgiving and she'll give you 20 bucks for that. Remind me again what the 3 types of cells that make up the juxtaglomerular apparatus are.
Granular.
Excellent.
Was it also macula densa?
Macula densa.
And then mesangial?
Excellent. Perfect. Remind me what each of these do. Blah, blah, blah. What does the macula densa cells do?
They have baro and chemoreceptors, and they also regulate, which allow them to regulate all of the like filtrate and stuff.
Close. Chemo and osmoreceptors. Remember, they're not ones that care about pressure. They're ones that care about the osmolarity. And they are found in what part of the nephron? Which part of the tubules? Which tubule portion? Remember, we want to be near the end of the process. So what's the farthest part of the tubule we can be in?
The distal
There you go.
convoluted tubule.
Excellent. So these are found in the distal convoluted tubule, and you're absolutely correct. They are monitoring the condition of the filtrate when it is almost unprocessed, being processed.
And they also release nitric oxide, right?
Excellent. And they release nitric oxide. Excellent. What do the granular cells do?
Dilate the smooth muscle for more or less filtrate to come in with the afferent arteriole.
Yep, I like that. Okay, so these change the diameter of what blood vessel?
The afferent arteriole.
Excellent. So not surprisingly, these are the smooth muscle cells of the afferent arteriole. Plus, they also have an endocrine function. They change the diameter of the afferent arteriole, which can increase or decrease blood flow into the glomerulus. Excellent. And that leaves us with mesangial cells. Where are they located and what do they do?
They control actin buildup or breaking it down, increasing the size of the glomerulus, excuse me, the surface area of the glomerulus.
Excellent, I like that. They contain accent and can change their size, increase or decrease. And as you mentioned, they're located within the loops of the glomerulus. And when they increase size, they expand the glomerulus and increase the surface area. And vice versa as well. They collapse, then they would decrease it. Excellent. So these were the three cells involved. So it all starts in the macula densa cells. So our macula densa cells, notice that the filtrate is overprocessed. So what is our problem at this point?
Filtrate is
Too
going
much infiltrate.
Too much or not enough?
Overprocessed. Too little.
Exactly. So our problem is we are making too little filtrate. or not enough. So macula densa figures that out. How does it resolve it? What does it do? You said it meant releases nitric oxide. So in this case, is it going to release more or is it going to release less?
More.
More nitric oxide. Excellent. What effect does that have on our granular cells?
Dilates the smooth muscle of the afferent arteriole.
And if we dilate the afferent arteriole, let blood into the glomerulus, and more blood into the glomerulus means more hydrostatic pressure, which means we make more filtrate, but we also what is the effect on the mesangial cells?
Build up actin.
Excellent. And when they build up actin and expand their side, they do what to the glomerulus?
Makes it bigger, increasing the surface area.
Increases it. So net effect of all of this is we have a higher hydrostatic pressure. We have an increased surface area, more blood and more surface area in the glomerulus. So that is going to equal, we make more filtrate. Problem solved. Any questions on that? Excellent. So then let's do the opposite. Macula densa cells, notice that the filtrate is under-processed. What is our problem here?
Making too much filtrate.
Excellent. So what do the macula densa cells do about it?
Release less nitric oxide.
Excellent. And what's the effect on the granular cells?
They don't dilate.
Excellent. And so because blood vessels have elasticity, they're going to constrict. The arteriole. So that means less blood into the glomerulus. Right, which means less hydrostatic pressure. What's the effect on the mesangial cells?
Break down actin, which makes the glomerulus smaller and less surface area.
Excellent. So our net effect, we have less blood and less surface area in the glomerulus. And so as a effect of that, we make less filtrate. And as a result of that, our filtration rate goes down and we have more time to process it. If you think about it, this whole page, I've done a ton of writing, but none of this is new. This is all the stuff that we talked about on what day is today? Tuesday. So on Thursday, when we talked about that juxtaglomerular apparatus and these 3 cells, we did this. So here we're just doing it again. And re-emphasizing that. And again, the beauty of both of these intrinsic processes is it allows us to auto-regulate every single one of those 1 million nephrons. Because when we send blood to the kidney, the same amount of blood's not going to go to all of the glomeruli. Not going to be at the same pressure. Some are going to be longer than others. Some are going to be shorter than others. They're going to have different filtration rates and processes. And each one of these can now be uniquely modified, both by how much blood is coming in them to begin with, but also by how successful they're being at filtering. So if it's a less efficient nephron, we want to have less blood going into it, so it has more time to efficiently clean the blood. So with both of these intrinsic mechanisms, we are able to, nephron by nephron, control the rate of the glomerular filtration rate, control how much filtrate is made. Questions on that? All right, excellent. Now, while we have all these local controls, we also want some big overlying controls as well. And those, as you guys already pointed out, are going to be our extrinsic controls, and that is going to be our hormones, and that is going to be our nervous system. Now, with our autonomic nervous system, as we mentioned, filtering the blood seems more like a housekeeping type of activity than it does a fight and flight type of activity. And so based on the type of activity it is, as we mentioned, it would seem to make more sense that this should be controlled by the parasympathetic nervous system. And surprisingly, it is not. It is actually the sympathetic. And now, even though I've said that to you a couple times now we can finally see why. Oops. Let's take a look at our glomerulus again. Nah, don't like purple. Here's our glomerulus. And as we know, we have that larger afferent arteriole coming in and the smaller efferent arteriole going out. And as we mentioned at rest, what percentage of your blood is being sent to the capillaries?
25.
Pardon me, sent to the kidneys, not capillaries, sent to the kidneys.
25.
Yeah, as we mentioned, about 25% of our blood is being sent to the kidneys when we are at rest. Notice that means we have a massive blood supply, which is going to give us plenty of pressure to do the work that we want to do. And while you're sitting here at rest and you have so much blood going to your kidneys, Is it necessary for the parasympathetic nervous system to tell the kidneys to do its job when it has this much blood coming to it? No, it's got plenty of blood, plenty of pressure, and it is very easy for it to do its job when we're sitting at rest. However, the problem we have is when we become active. When we become active, right, our fight or flight, or just getting up and exercising, or running downstairs during the break to get a cup of coffee or whatever it is, as we know, when we become active, much more, well, let's say this way, more blood is redirected to other parts of the body, like the liver, like the lungs, like the skeletal muscle. And as a result of that, much less blood travels to the kidneys. And that's a problem. If we have less blood in the nephron, in the glomerulus, means less pressure, means less filtrate. And as we talked about before, so every time I get up and run downstairs to get a cup of coffee during the break, do I want to be hurting my kidneys' ability to be able to filter blood?
No.
No. So what we need is a way to modify the pressure in the glomerulus to compensate for having much less blood. And that's what our sympathetic nervous system does. Our sympathetic nervous system helps our glomerulus. Oops. So it helps us to maintain normal filtration rate. And the way it actually does this is our sympathetic nervous system, and I'll just abbreviate that SNS here, causes both the afferent arteriole and the efferent arteriole to constrict. Now, let's think about those effects. When the afferent arteriole constricts, what does that do to the blood inside of it?
Increases the blood pressure, kind of constricting the amount of blood that goes into the glomerulus.
Well, notice it isn't necessarily constricting the amount of blood, but you're absolutely right that this increases the pressure. Let's say I'm out in the backyard watering the garden, and my wife walks outside, and the stream from the hose isn't enough to reach her. But being the loving husband that I am, she looks very hot, and I want to spray her with water to help her feel better. So what can I do to make the water go farther? What can I do to increase the pressure of the water?
Put your finger over it.
As you put your finger over the opening, you decrease the diameter, and that increases the pressure. Notice we have less blood coming in, but this increases the pressure of the blood that's there. And that's going to help us to compensate. So notice we have a smaller volume of blood. That's the problem. Our problem here, let's say it here, we'll mark it up here to remind us. Our problem is we have a smaller volume of blood in the glomerulus. So one of the ways we fix that is by constricting the afferent arteriole. That increases the pressure of the blood that enters the glomerulus. But remember, it also constricts the efferent arteriole. What does that do? What does constricting the efferent arteriole do? All right, let's think about this. You've got a holiday on Thursday, so you don't have class again till Tuesday. Perfect opportunity to fly to go down to Disneyland for a few days. Right. When you're coming back, and it doesn't matter what time you're coming back, you could be coming back at 4 o'clock in the morning on a Wednesday. You're going 105 miles an hour down 80, and then you hit Davis. And what happens when you hit Davis, even at 4 o'clock in the morning on a Wednesday? Traffic slows down. Why? Why does traffic always slow down in Davis?
There's less lanes.
Exactly. Notice you go from the 10 lanes that there are of 80 down to 2. And when you go down to those 2 lanes, you produce congestion. And congestion produces turbulence. Notice this is going to keep the blood in the glomerulus and swirling around in the glomerulus for a longer period of time. And if the blood is in the glomerulus longer and bouncing on the walls more because of that turbulence, what happens to the pressure?
Increases.
It increases. Absolutely. Hold on, Ruby. I see your question. Let me finish writing this down and then I will answer it. So this, let's say it this way, reduces, or let's say it this way, slows the blood from leaving the glomerulus and produces turbulence in the glomerulus. And both of those things increase the pressure in the glomerulus. So notice by both constricting the afferent arteriole and constricting the afferent and efferent arterioles, we are increasing the pressure. So even though we have less blood now in our glomerulus, we can maintain the right pressure to do work. Yeah, so true, you are correct. By constricting the efferent arteriole, it does lower the pressure of the blood as it leaves the glomerulus. But remember, as it leaves the glomerulus, this is going into the paratubular capillaries. And remember, those are typical low pressure capillaries. So we want the pressure to be lower. Remember, we want the high pressure in the glomerulus. We want low pressure in the paratubular capillaries. So yes, as the blood leaves, it's at a lower pressure, but that's a good thing. We want it lower. So great question. I love that. Excellent. Perfect. So questions on this?
Yeah, I'm sorry. So the overall effect is trying to keep the glomerulus from, is trying to maintain the right pressure?
Yep.
Is that what I understand? Okay.
Right, because remember, to produce filtrate, we have to have a higher outward force on the blood than the inward forces. And the only outward force is that hydrostatic pressure of the blood. So if our blood pressure drops in the glomerulus, we can't make filtrate. And the
Okay.
problem when you're exercising is much less blood is going to the glomerulus. And if you have less blood, you have less pressure. So this allows us to increase the pressure of the smaller amount of blood coming in and keep that smaller amount of blood in there for a longer period of time, which will give us the appropriate pressure, help us to maintain the pressure even though we have less blood. We're still able to get the right amount of pressure even though we have less blood in the glomerulus, and that allows it to continue to do its normal job.
Thank you. I appreciate it.
Yeah, no, it's a great question. Any others? Now, there's one more point I want to make on this. When that bear with an axe walks into your room, and you really do have to either run for your life or fight that bear. In that incredibly high stress situation, is the sympathetic nervous system going to be able to constrict the afferent and efferent arterioles enough to maintain appropriate pressure to deal with, to filter blood properly in the kidneys? What do you think? No, exactly, right? So when you're in that high stress situation, like that bear with an axe, this is not to compensate. And so when you are in that high stress or high energy, right, your kidney function does decrease. Now, if you are fighting for your life with a bear with an axe, is a decrease in kidney function your biggest concern? Not a trick question.
No.
No, absolutely not. Or if you are sprinting in a race, right? Is the lack of kidney function or decrease in kidney function your number one concern at that point? No, you want to win that race. However, if you're sprinting, how long is that race lasting? Yeah, 10, 20, 30 seconds, maybe a minute. That fight with the bear with an axe, how long is that going to last? Not very long, exactly. So yes, there is a minor decrease in kidney function, but typically these are very short-term events. However, remember someone mentioned in the last class about how chronic stress, if you have that long-term chronic stress from a job that you hate or a bad relationship or an A&P class where your instructor's a jerk, in any of those types of situations, when you have that prolonged stress that lasts for a prolonged period of time, is there a potential for that to damage the kidneys long-term? Yeah. And so that stress, that long-term chronic stress can be an issue for kidney function. I know someone talked about that or mentioned that in the last class. And so now we see why. The sympathetic nervous system can try to compensate some, but with chronic stress, with that long-lasting high level of stress, it can actually be damaging to the kidneys. They'll lose their efficiency and function. All right. Questions on that? All right, excellent. So again, our sympathetic nervous system constricts both the afferent and the efferent arteriole. And since the afferent arteriole is surrounded by those juxtaglomerular cells, those granular cells, our sympathetic nervous system can also stimulate the release of hormones. Now, the good news is 2 of these 3 hormones we have already talked about pretty much in a fair amount of depth. Antidiuretic hormone. All right, small, so it all fits in here. Someone remind me again where that is produced. If only I'd learned my hormones, I'd know what organs, what glands were responsible for producing my individual hormones.
Posterior pituitary.
Excellent. Well, although technically it's produced not in the posterior pituitary, technically it's released. So you have to even trick myself on that. Where is it produced?
Oh yeah, the hypothalamus. I forgot about that.
I did too. Notice I started writing it wrong as I was doing it, but then I was like, oh, wait, hold on, catch that. Let's talk function. What did we say antidiuretic hormone did?
It makes your body keep more water. So because the diuretic makes you excrete more water. So if you keep more water, then it would increase the blood pressure.
There you go. So it's going to cause us to reabsorb more water and also some sodium as well. And of course, at the time, we said the target was the kidney. However, now we can be a little bit more specific. In this case, as it turns out, the target, remember, as I mentioned, where we do that hormone determined fine tuning is in the distal convoluted tubule and the collecting duct. So in the collecting duct, in particular in the collecting duct, remember the collecting duct we said has two types of cells. It is the principal cells of the collecting duct and the distal convoluted tubule, where that antidiuretic hormone tells those cells, bring back more water. Excellent. Oh, and look, we have another one here that we learned about. Atrial natriuretic peptide. Where is that produced?
The heart.
Although we can be a little more specific than that.
Atrium.
Yeah, I was gonna say, you're overthinking it. Atria, absolutely, excellent. What is its function?
Does it regulate the blood pressure of the blood going into the atria?
Yeah, so remember when the blood pressure was too high, we needed to bring that blood pressure down. So its function was to lower blood pressure. And how did we get it to lower blood pressure?
Were there baroreceptors?
That was what, so that was the sensory structure that noticed there was a pressure change, produced the atrial natriuretic peptide, which then went to the kidneys and tells the kidneys are target here, to release less water, or more specifically, to reabsorb less water. And when we reabsorb less water, that lets more water leave. So notice antidiuretic hormone tells the kidneys to bring more water back. Atrial natriuretic peptide told the kidneys to let more water leave. And not surprisingly, this is also going to be in the distal convoluted tubule and the collecting duct. Because again, that's where our hormone-based fine-tuning occurs. So even though that one was a little like pulling teeth, these were things that we have talked about before. So again, we did that, produced by and released by, tells the distal convoluted tubule and the principal cells to reabsorb more water. As you mentioned, by bringing more water back, that is going to increase blood pressure and less water in our urine. Atrial natriuretic peptide, remember, is produced and released by the atria of the heart and tells it to release more water, lowering our blood pressure. So again, those are the ones we've talked about before. What we mentioned before, but we didn't talk about, and now is going to be our chance to talk about it more in depth, is our renin, angiotensin, aldosterone system. Renin, remember, as we mentioned, is a hormone-like protein that is produced in the kidney. specifically by those granular cells. They are measuring the blood pressure and therefore the blood volume of our blood. And if that blood pressure or blood volume drops, our granular cells produce renin. Now, as I said, it is not a hormone, but it likes to pretend it's a hormone. And one of the things that it does is it activates a plasma protein called angiotensin. Angiotensin 1 is actually produced by the liver. However, there is a special enzyme found in the lungs. And that enzyme that is found in the lungs converts it into its active form. So basically, angiotensin 1 is an inactive form. It's a plasma protein produced by the liver, but when it runs into that active enzyme in the lung, it becomes activated. And that active form is angiotensin 2. And angiotensin 2 does a whole lot of stuff. All right. Questions on this before we talk about what angiotensin 2 does? This starts in the kidney, activates a plasma protein called angiotensin 1. That protein is made in the liver. And then in the lungs is a special enzyme that converts it into its active form. Notice, this is a pretty elaborate system, but if you think about it, the kidney, the lungs, the liver, these are all big places where a lot of blood goes to. So I know it may seem a little random to have it all spread out, but these are all three very important locations where blood is going to travel to. So it does kind of make some semblance of sense, hopefully. All right, angiotensin 2, this is our hormone. And like many hormones, it has a lot of effects. So let's talk about the effects and think about what they're going to do. One of the primary things that angiotensin does is constrict our systemic arterioles. Pretty much systemic arterioles are found over most of the body. And if most of our systemic arterioles were to constrict, what effect would that have?
Increased blood pressure.
Increased blood pressure. Excellent. Angiotensin 2 targets the kidney. Again, the distal convoluted tubule of the nephron, and tells it to reabsorb more sodium, more chloride, more water. And if we're bringing more salt and more water back into the body, blood volume goes up. And of course, when blood volume goes up, what happens to blood pressure?
Increases.
It increases as well. Excellent. Angiotensin 2 triggers aldosterone to be released. What part of the adrenal cortex produced aldosterone again? Give you a hint, it starts with zona. Don't you hate it when the instructor asks you to remember things that you're supposed to remember in a cumulative class? That was like 2 sections ago. Who remembers endocrine system anymore?
Is it reticularis or fasciculata, something like that?
So there was glomerulosa, reticularis, and fasciculata. Which one was it? Anyone?
Isn't there
The zona glomerulosa, right? The ball-shaped ones. And all right, so what does aldosterone do? What's it target?
The gonads.
No, aldosterone didn't target the gonads. Aldosterone targeted, well, which organ system are we in?
The kidneys.
There we go. And it targeted the kidneys, telling the kidneys to reabsorb what? Remember, aldosterone was our most common mineral corticoid, which helps us to regulate our electrolyte levels. And what's our most common electrolyte?
Sodium.
There we go. And of course, where sodium goes, what's going to follow?
Water.
There you go. So if the kidneys, and again, this is going to be in the distal convoluted tubule and the collecting duct, is bringing back more sodium and more, well, water follows it, blood volume goes up, which increases our blood pressure. All right, I'm gonna have to make that small. Sneak it up here. Angiotensin 2 stimulates your hypothalamus, making you feel thirsty. And what do you do when you're thirsty? You drink, meaning that there's more water absorbed by your digestive system, mostly in your small intestine, which means you have more water in your blood, which does what to blood pressure?
Increases.
Increases. And because, again, all of these systems help themselves, this also stimulates the release of antidiuretic hormone, which also causes more water to come back. So notice all of these factors do one of not two things. They increase the volume of blood and they increase blood pressure. Right. And if you remember, the trigger was a drop in blood volume, a drop in blood pressure. So this hormone helps to bring it all back up. Because as we've seen, even small changes in blood pressure can have a big impact on the function of the renal corpuscle. Here we've done it with all the pretty words, but your book's got this great illustration that kind of shows all of the same stuff, right? We have a decrease in blood flow to the kidneys. So that is noted in our granular cells. They release renin. Renin causes angiotensin to be produced and causes it to be activated into angiotensin 2. Constricts the efferent arterioles, increasing the pressure in the glomerulus, increases overall peripheral constriction. Here we go. Our systemic blood vessels, which increases pressure in the glomerulus. Right. It stimulates our sympathetic nervous system, so we get more heart beating faster, right? Bringing more blood back, getting more water, increasing blood volume, increasing blood pressure, bringing everything back. So we have all of this, bringing back more sodium. All of this stuff is here. There's this beautiful little flow chart that goes through all of it on this renin, angiotensin, aldosterone system. All right. Questions on that? All right. Excellent. Now we have talked about glomerular filtration, the first step in this process. We've talked about the pressures that produce the filtrate, how much filtrate we want to make, how we regulate and monitor how much filtrate we want to make. You have now learned everything you ever wanted to know and more about glomerular filtration rate. And again, because your book likes illustrations, here's another nice flow chart that puts all of these together. And notice the beauty of this is it shows all of those controls we were just talking about. Our intrinsic controls and the two mechanisms, both the myogenic and the tubular glomerular feedback, our hormone, and our neural control. Puts it all together with that same ultimate goal, right? If our systemic blood pressure drops, we need to bring it back up. We need to maintain glomerular filtration rate. So we've learned everything you wanted to know and more about making filtrate. So now we can switch gears to our other two processes, tubular reabsorption and tubular secretion. All right, we can finally move from the renal corpuscle into the proximal convoluted tubule and beyond. Now, before we do that, let's go ahead and take one more break because that was a lot of information. It is 2:15, so we will restart at 2:30. And I will start the recording at that time. Any questions before we take our second break? All right, I will see you guys in 15 minutes.